Sounds. Music, radio, podcasts.
Hello you, this is the podcast of Inside Science from BBC Radio 4, first broadcast on the 12th of March 2020. I'm Adam Rutherford. This week's programme is a coronavirus special. How is it that something so very tiny has caused a global medical and economic crisis? We're focusing on the virus itself, what it is, how it evolved, host animals, and how our own behaviour caused the spillover from animals into us. Dealing with an infectious disease of this scale is a complex ecosystem of medicine, of behaviour change, of policy, and many other things. And other Radio 4 programmes have devoted their airtime to different aspects of Covid-19. Inside Health and more or less coronavirus specials are all available on BBC Sounds. But all of the policy and medical interventions rely on high-quality science, and that is what we are all about here on Inside Science. Virologist Jonathan Ball from Nottingham University has been our guide for many weeks now, and just to get us right back to basics, we asked him to describe exactly what the coronavirus actually is.
It looks like a crown, so it's a round blob, and projecting out of that blob are various protein spikes. And those spikes are what the virus uses to allow it to get into the cell. Those spikes are embedded in a layer of what we call lipids, and this is a kind of oily layer. And it's that oily layer that makes the virus vulnerable to things like alcohol rubs and soap and water. But essentially that virus has to get into a cell, and it does that in the respiratory tract, so your nose, your throat or your lungs. It attaches to the cell using those protein spikes. That allows it to then gain entry into the cell, and once inside, it uses its own proteins, its own enzymes to replicate itself to make lots of new copies of virus, which then go on to infect other cells and unfortunately go on to infect other people.
Now, when you were describing that, how the virus enters the cell, so some viruses sort of inject their genetic material, either RNA or DNA, directly into the cell. But in the case of coronavirus, what you're saying is that the whole virus itself inserts itself into a cell.
So once the virus has attached to the cell using those protein spikes, what it then has to do is get into the cell, and it does that by allowing its oily layer to actually combine with the membrane that sits on the surface of your cells. And then once that fusion takes place, the virus is inside the cell, it can then start to copy itself.
And we'll be coming back to Jonathan Ball later in the programme when we'll be talking about the process of developing vaccines. But first, viruses evolve, just like all living organisms, via genetic changes every time they reproduce. This means that we can track and trace the way a virus changes, how it adapts, and the mechanisms by which it becomes infectious by reading the genetic code of viruses taken from infected people. Charlotte Houldcroft is an evolutionary virologist at Cambridge University, and I asked her to tell me how they get from patient to DNA sequence.
If you take a swab from someone who's infected with the new coronavirus, the coronavirus genome is about 30,000 base pairs, or bases in length, so it's much smaller than the human genome. And that swab is going to have human DNA from cells that have been scraped away and all the other viruses and bacteria that live in your nose and throat. So we have to use techniques like polymerase chain reaction to amplify the amount of viral genetic material that's present before we can go on to sequencing it, or we have to sequence it in very high depth, which takes longer and is more expensive. So most labs in the UK will be amplifying, increasing the amount of viral genetic material that's present before they start to sequence.
But you have to sift through a lot of DNA and RNA in order to get meaningful sequence from just the virus.
You do. You might find that only 1% of the data that you acquire is actually what you're interested in, depending on the method that you use. Although something like PCR increases your success rate to more like 80% of what you're looking at is what you're interested in.
And in terms of the sequencing itself, the accuracy of the actual letters that you get out of the virus, so we're confident that the sequencing is accurate and informative.
New sequencing technologies sequence each individual base of the virus thousands and thousands of times, and that means that even if there is an error rate in the sequence data, because you've effectively read each letter 1,000 times, you know that the majority letter at each position is the correct one, and so we can be confident that when we do see a mutation, that it's real.
Obviously, the new coronavirus is new, and therefore we're learning about it as we're going along. In your work, what can we learn about how diseases spread, how viral diseases are transmitted from animals to humans or through human populations by looking at the evolution of their genomes?
So we can look for how fast differences appear, so that's the mutation rate, and we can also study specifically where in the virus genome those mutations occur. And we know for coronaviruses that mutations in the spike protein, which is the way that the virus gets into the cell, are very important for its transmissibility, and they're also associated with its virulence, which is how likely it is to cause symptoms.
And those mutations in that particular gene, do they change at a specific rate, or I mean, what is the sort of selection criteria for seeing that evolutionary change over the generational time of a virus?
The novel coronavirus has a proofreading enzyme, which means that it doesn't make mistakes as often as, for example, HIV or other viruses you might have heard of. So we don't see a mutation in the genome every time it transmits from one person to another. And actually, one person might infect another person with exactly the same strain that they were infected with. And something like 1 in 20 transmission events creates a mistake that's passed on. And only a very small proportion of these are going to be within the spike protein that matters. And we haven't yet seen any within the sequence diversity that's being seen worldwide, but we can look at the differences between the spike protein of the novel coronavirus and its relation, the SARS virus, and we can see some differences which might be making it easier for this virus, this novel coronavirus, to get into cells within the lungs.
Does that also make it easier to track the fact that there is a lower mutation rate or this copy editing enzyme that actually corrects mutations?
Not really. Unfortunately, it's making life more difficult because it means that when you see two cases with an identical sequence, one person could have transmitted directly to another, but you could also be missing a third link, a third person who transmitted to both of those individuals, or an animal reservoir that transmitted to those two individuals. And you can't prove it with the sequence data alone, though it can really help if you combine it with things like contact tracing.
Right, because in the last few weeks, I mean, it seems like a lifetime ago in the course of how fast this story is changing. But there were the cases where where people were being infected, and we couldn't see obvious transmission routes, people-to-people contact from, for example, from China. So, can DNA? Can the genetic sequencing of the virus in these patients, can that help us understand that transmission, which is a bit of a mystery in terms of contact?
It can actually be most useful in ruling out direct person-to-person transmission. For example, if you look at the sequences that have been made available from Scotland, we can see there's been 2 separate introduction events. They have so many different mutations between them that they can't have come from a common source, and that gives you some confidence that the tracking and tracing is working. But actually, sometimes you can get false positives from looking at the sequence data alone, where you might infer that 2 people have been into contact, or that there's a transmission chain going on within a country because their sequences are very similar. But actually, you can get exactly the same patterns in the sequence results if they both come from the same source somewhere else. So that might be the same ski resort, it might be the same pilgrimage, it might be touching the same contaminated surface in an airport.
And is this what we're seeing when in the recent reports just from a couple of days ago that cases in Italy had been derived from a Bavarian infection because that's what the initial sequencing looked like, but it turns out that may not be the case after all?
Yes, that's exactly what has happened, that the story that the virus might have transmitted through Europe from Bavaria to Italy is one way of explaining what we see in the sequence data, but actually a much simpler explanation is that 2 travellers, for example, from the same part of China several weeks back, may have transmitted identical strains to 2 different parts of Europe that have then seeded different hotspots. It doesn't necessarily prove that one hotspot in Europe led to another hotspot in Europe. They could both be coming from the same external source elsewhere in the world, and we cannot distinguish between the 2 with the virus sequence data alone. We have to have classical epidemiology.
Okay, but the sequencing though, so this is your speciality, but are you saying that sequencing as many samples as possible from as many different people in as many locations is actually going to be essential, a sort of bedrock from which we can really try to understand how the disease is spreading and evolving?
That's definitely what I feel. I think this is the first outbreak where we've been able to sequence virus samples in real time. So turnaround time of something like 8 hours from sample to genome sequence, which has just not been possible until a few years ago. And it's allowed different parts of the country to see in real time whether their infection control is working, that you can see that different cases that are occurring within Wales, within Scotland, come from separate introductions. And that tells the public health staff that what they're doing is working. And that's really powerful information for us to have.
Charlotte Houldcroft from Cambridge University. It's important to remember that this virus is only new to us. We have not evolved alongside this particular coronavirus, and so it presents a serious threat to our own immune systems. As with many pathogens, they exist in animal populations where they may or may not cause disease. But it is the pathogen itself that makes the jump from its animal host, what we sometimes call a reservoir, to us, and that almost always happens as a result of human behaviour. The origin of the current coronavirus is not fully known, though since the beginning it was presumed to have emerged from a wet market in Wuhan in China, with various animals on sale under the catch-all title of bushmeat. This includes bats, which we know also act as a reservoir specifically for coronavirus, though bats do not suffer the same symptoms as us. Kate Jones from University College London is a bat expert and a zoonotic disease expert, and I asked her if there was anything special about bats and their evolution alongside coronaviruses.
It's a qualified yes. So bats have evolved to fly, clearly, and because that's very metabolically active and expensive thing to do, so sometimes it's like 34 times as their base rate when they're flying, their metabolic activity rate. That's an incredible amount of pressure to put your body under. And so there's some thoughts around how adapting to that metabolic stress and oxidative stress has enabled viruses to co-evolve with bats. Bats are much more, they seem to be much better at dealing with oxidative stress and DNA damage and repairing that damage. And it also means that they are pretty resilient to having these persistent infections without getting sick. So that's not to say that they don't get sick, but it's just seen that like RNA viruses, they can tolerate them, so they can deal with the infection and it can persist. They don't necessarily have to clear it to be able to survive that infection. And it's only when perhaps they're metabolically stressed or food stressed or in pregnancy or lactation that they may have to mount an immune response to some of these viruses, and then they may be shedding these viruses in a higher frequency. So it could be that during those times they're more infectious to other species.
Well, let's talk about how it gets from bats to us, and obviously we don't want to just attribute bats as being the source of this problem. There is a process by which a pool of viruses in a particular group of animals makes it to us. How do we go about understanding that process?
So there are many kind of ways to think about this, but it's largely about human behaviour and human behavioural change, which impacts this transmission dynamics between bats or any wildlife host of a pathogen and humans. So it's about how the transmission is changed with human behaviour. So, for example, we're moving animals and trading animals, pet trade and for eating meat than we have ever before. So we're moving animals more than we ever have. We're doing things at a rate and a scale that we've never done before. We're mixing species into wet markets. We're combining things together which have never been seen before, and these are important things because our human behaviour is affecting the transmission dynamic. So something that we would never have come into contact with, it lives happily and co-evolved with a particular wildlife species, then our behaviours are changing those transmission pathways. So it's not the species' fault that we're getting these diseases from it. It's our behavioural changes which are impacting the transmission dynamics, which is changing that.
And even though from the very beginning we've been talking about a particular wet market in Wuhan city, where we think it originated. Have we got any direct indication that it was a particular type of bushmeat? Was it even the presence of bats as bushmeat in the market that we say was the pool from which it jumped to humans?
Yeah, we don't know. I think we've got similarities of the strain from known viruses in some bat species. It's probably mixed with lots of other coronaviruses in other species, and then you have a mutation. So, I mean, the direct evidence is still quite vague at the moment about what species it was, and even if it was bats, I hate this demonization of any species when it's clearly our fault. It's our behavioural changes which have caused this. It's not the wildlife fault that dragged it into a wet market and its pathogens have combined together to cause an outbreak. It just gets me really angry.
There is some emerging evidence that there may have been an intermediate mammalian species, something that we don't know, between bats and humans. So have you got any thoughts on what that process might have been and what that intermediate species is likely to be based on our changing behaviour?
Well, it's about how we're moving animals and trading animals and using animals as bushmeat into these markets, and so that you may have crates of animals on top of each other which never would have come into contact with each other normally, and that's a perfect storm for pathogen mixing between different species which would not be in contact with normally. So I think it's a bit preliminary to pick on a particular species, but I'd seen a report of pangolins, and pangolins are one of the most traded animals in the world, so it could possibly be that, given their presence in a lot of these markets.
How important is the identification of the reservoir species for understanding the disease and how it spreads and just how we tackle it? Do we need to know the pool that it came from?
I think it's really useful to understand the ecology and the ecological systems within which these pathogens are present, because we need to understand how our actions are going to affect the prevalence of this disease and their impact on the host populations. So unless we understand how the host is responding to global changes like climate change, ecological systems changes like deforestation or bushmeat hunting, we won't know how that pathogen might react under different future scenarios. So I think the ecology is really key. So knowing where that virus is, what species it's in, but really importantly, how that species responds to global changes, I think is really important.
Kate Jones from University College London. This is Inside Science, a coronavirus special. Now, understanding the basic biology is essential for developing a vaccine. The Jenner Institute at Oxford's Nuffield Department of Medicine is a key UK centre for vaccine development for a whole range of diseases, but they have effectively dropped much of their current work in order to focus almost exclusively on coronavirus vaccines. Professor of Vaccinology Sarah Gilbert at the Jenner explained the necessity of a coronavirus vaccine.
Well, we know it's something that is spreading very rapidly now, and we are going to need to have a vaccine against it. Now, nobody is promising that there will be a vaccine in time to stop the spread of the disease in any country in the world. It takes longer than that to develop a vaccine. But we know that this virus isn't just going to disappear the way that SARS did. So in 2002, there was outbreaks of the SARS virus. It rumbled on for a few months. There were cases and deaths, and then it was contained, and a vaccine was never developed. This has spread much further, much faster. What everybody's now expecting is that even if it is contained, it's quite likely to come back because it's spread so widely now, and it may not be contained. It may be something that goes through the population and becomes part of the panel of viruses that causes respiratory infections every winter, and we then have to think about whether we want to be routinely using a vaccine against this particular coronavirus, maybe in the high-risk populations, or whether it's something that can be managed in some other way. But it's likely that for at least some of the population we'll want to be able to vaccinate them in the future.
And Jonathan Ball told us how vaccines actually work.
Essentially with a vaccine, what you're trying to do is get your immune system to recognize and remember a viral infection. And what you hope that happens is that you either develop antibodies, and these antibodies are proteins that your blood cells make, your white blood cells make, and they attach to the surface of the virus and then stop it interacting with the cell. And so they neutralize the virus, they stop it and kill it. But another aspect of your immune system, it produces what we'd like to think of as killer cells. So these are cells which are primed to recognize any other cell in your body which is infected with a virus, and then they'll go along and destroy that cell to prevent all the virus that's being produced inside that cell from leaking out and then going on to infect other cells in your body. So vaccines can work in a multitude of ways, but essentially what we're trying to do is get your immune cells to recognize and remember the virus, so that when you encounter it, it can release these killer antibodies or these killer cells and destroy the virus in its tracks.
And what are they looking to recognise on the surface of the virus itself? What is the antigen, the thing that they're trying to lock onto?
It all depends on which part of the immune system you're trying to provoke. If it's antibodies, then what they'll do is look for the proteins that are displayed on the surface of the virus, so the spike protein. They'll attach to that protein and they'll stop the virus getting into the cell. But for these killer cells, they can recognize a whole host of different viral proteins, because as the virus replicates inside a cell, some of the proteins that it's producing that are viral specific will be displayed on the surface of the infected cell. That's a red flag to the immune system, and they'll send these killer cells in, home in on it, and destroy the cell.
So in terms of developing effective vaccines, plural, how many different sort of strategies can we employ? Will different labs be doing different things?
Well, that's the advantage, and that's the key here, is because we can't predict exactly which vaccine strategy will work. So some labs will be focusing primarily on producing protective antibodies. They might be using just the spike protein of the coronavirus to try and train your immune system to recognize it. Others might be just interested in these killer cells, so that they'll be using a variety of viral proteins to try and get those killer cells to recognize it. And then others might be interested in both arms of the immunity, and they might be using things like live vectors. So these are other viruses which have been genetically modified to carry parts of the coronavirus into the body. And then when those viruses are introduced, the immune system recognizes the coronavirus parts. It trains your immune system, and then when you become exposed, or when you get exposed to the virus, they go in and kill that virus.
And this is exactly what Sarah Gilbert's lab is doing right now to develop a coronavirus vaccine, though it involves fusing it with another well-characterised virus.
Well, what we do is use molecular biology initially. So we have the genome of the adenovirus that we use to make our vaccine, and we've added some extra sequences to it so that we can then make more copies of it in bacteria. So it's now a bacterial artificial chromosome, a large piece of circular DNA that we can put into bacteria, and as the bacteria grow, we make more of this bacterial artificial chromosome, and then we can purify that DNA and we can manipulate it to add in a gene that encodes part of the novel coronavirus, for example, that we want to use to make the vaccine. And then when we've done that, and we'll do some testing on it to make sure it's the right thing, we take that bacterial artificial chromosome, which contains the whole genome of the adenovirus plus the gene of the novel coronavirus, just the single gene, and then we put that into mammalian cells, and that starts to form the adenovirus vector vaccine. So now we've got live adenovirus being formed in the mammalian cells. It can only make more copies of itself in that particular cell line, so it doesn't do that when we use it as a vaccine, but that's the process that we use to increase the amount of virus that we have, and then we have to purify the virus away from the cells that we've used to produce it on. So we end up with a very highly purified preparation of the adenovirus with the novel coronavirus gene in it, very concentrated, and with no other pathogens, no other organisms present in the vial, and that is then essentially the vaccine.
Vaccine development is ongoing, but is unlikely to deliver during this current wave of the pandemic. The WHO yesterday declared that it is a pandemic, and that means it's not going away easily or soon. There are, however, other drug options for treating the disease and, crucially, for preventing the virus in the first place.
So there's a variety of compounds that are being actively explored. Some of them are anti-HIV drugs, and what researchers have done have used structural data, so they know what these proteins look like, and they know the business parts of these proteins, what we call the active sites. And they're able to look at great detail at molecular level to try and model how drugs might fit into those, what we call the active sites, and stop the protein, the enzyme from working. So that's one aspect that's been actively pursued. Others is to repurpose drugs that prevent the enzyme that copies the genome of the virus. So these are so-called polymerase inhibitors. Remdesivir is a well-known example, and that's being trialled as well. That was developed for Ebola, but it's now being repurposed, hopefully, for coronavirus. So what they do is they try a variety of different antivirals. You then can test them in a lab, see if they can inhibit virus being grown in a flask. If they do, and you have an animal model, you can then see if they protect animals, and then hopefully take them into human trials.
Okay, now just to finish off, Jonathan, we've been talking to you since the very beginning of this for many weeks now, and we're very grateful for your expert advice on this whole subject. It has escalated at a rate that I'm not sure anyone thought it was going to go in this particular trajectory. But how important at this point is understanding the basic science compared to actually containing the disease spread via policy and behavioural change?
It's hugely important because, funnily enough, understanding the basic science can have a massive impact on the policy around trying to control this virus. So, for example, we don't fully understand at the moment just how many people do have very mild or even asymptomatic, so symptomless infections. We suspect that lots more people have been infected than have been recorded, for example, in China. And when you have that data, you can start to get a better handle on just how deadly the virus is, how many cases really do result in serious disease and death. And so I think, that there are some key basic scientific questions that still need to be addressed, and over time we hope that we'll start to gain an insight into that so that we can start to inform policy and also predict just how this epidemic and pandemic will develop over the next few weeks, months, and even years.
Virologist Jonathan Ball from Nottingham University. Understanding the basic biology and evolution of this virus is the bedrock to any medical or policy interventions. Without decent science, policies are built on sand. Thank you to all our experts, Kate Jones, Charlotte Houldcroft and Sarah Gilbert. That is all for now. Email us, BBC Inside Science at bbc.co.uk. This is Inside Science, produced in the BBC Radio Science Unit by Deborah Cohen. I'm Adam Rutherford. See you next week.